Shall we begin? 各位来宾、女士们、先生们，早上好。Dear honorable scholars, distinguished guests, ladies and gentlemen, good morning. My name is Wening Zheng. I'm the chair of South Genomic Research Center and also professor of South Medical University in Guangzhou, China. It is my pleasure to hold this opening ceremony of the World Congress Gene 2008. After nearly 1 year's preparation, under the great efforts of organizing committee members and our respectable SAB members, the second annual World Congress of Gene 2008, hosted by China Medicinal Biotech Association, People's Government of Sanshui, Fushan Government, and organized by People's Government of Sanshui District Science and Technology Bureau of Fushan City, and also by Dalian BIT Life Sciences Incorporated China, and supported by many other organizations, now is opening.
Thank you very much. Well, my talk is going to be primarily about translational medicine that Dr. Hershko mentioned as being one of the waves of the future. I'm going to be talking at the level of the gene, and I'm going to be talking at the clinical level as well. And I would like to first make an apology to those of you who are scientists here for explaining what in silico means. But there are 3 phases of biological research. There's in vivo, that means in living persons. There's in vitro, which means basically in the lab, in cell culture. And there's in silico. The first time I came across in silico research was here at the Hospital for Sick Children in Toronto back in 1981. This is a group of us there at the time. We'd just come from a conference where IBM had shown how they were using their new supercomputers to produce the human insulin. This was the Humulin, and it was synthesized with the use of in silico technologies. It was primarily done by computers, and it was one of the first of the drugs that were the major drugs that were produced by computers. This was my first introduction to in silico work. But most of you would be more familiar, of course, with the use of in silico technologies in the decoding of the human genome, and perhaps more importantly, although less well known, in the decoding now of 1078, last time I looked it up, 1078 bacterial genomes are now fully decoded. And the NIH has just started a large human microbiome project which over the next 10 years aims to define exactly what microbes exist in the organism we called Homo sapiens. Basically, you'd all be familiar with the normal areas where microbes are expected: nasal cavities, oral, skin, GI, and urogenital. But it's known that microbes are far more prevalent in the human body than anybody up until now has even dreamed. The NIH says that in a human body, approximately 10% of the cells are human cells, and approximately 90% are bacterial cells. Now remember that bacterial cells are really small, so you will have hundreds or thousands of them inside each infected human cell. However, those are the numbers from the NIH microbiome project, and that's a staggering start. We then go to the next set of numbers. There are approximately 25,000 human genes active in the human body, and about 1 million bacterial genes. The interaction between these bacterial genes and the human genes, of course, will help define how the organism Homo sapiens actually operates. Well, why is this important? If we look at, for example, the E. coli glucose metabolism, you can see at the top here you've got glucose 6-phosphate coming in, and at the bottom I've taken it down as far as pyruvate, citric acid cycle down below. But basically this shows each of the genes in the E. coli organism, the proteins that are produced, and then the end products of that protein action on the input glucose 6-phosphate. And the main thing to note is it's not all that much different from the way the human body utilises glucose. You can see a lot of names that you recognise. Obviously the end products here, adenine, serine, cysteine, the amino acids, and also tyrosine, tryptophan. Those are very well known. They're invariant in man. But you'll find also glycogen and a number of other ribose 5-phosphate, a number of other metabolites that are very common if one studies the human glucose metabolism. This particular analysis was done by Vijay at Bielefeld University in Germany. And he has done a lot more work than just this one slide shows. But really, what I wanted to point out was there is not a lot of difference between the way that E. coli metabolizes glucose 6-phosphate and the way the human body does. Many of these metabolites are the same. And if you could have an interaction between the E. coli genome and the human genome, then an entire new spectrum of transcription and translation is opened up. Now, in fact, published in Proceedings of the National Academy of Sciences last year, some Kwang Il-go and al. actually took the gene bank, and they took all the genes in the gene bank, and they then plotted all of the diseases, bone, cancer diseases, right through to skeletal, respiratory, renal, and they plotted from the gene bank all the various diseases and the genes that were common to the various diseases. Now, if we enlarge upper section of that, we have the rheumatic diseases here. The rheumatic diseases because, in fact, those of you who are from Sichuan province, I'm going to be giving a more extended seminar next Monday at 2 p.m. at West China Medical Center in Chengdu. So I'm focusing on the rheumatic diseases here, but we could focus on the neurological or any of the other diseases. If we take this one gene here, ACE, which produces the angiotensin converting enzyme, it is known to be associated with myocardial infarction, with renal tubular dysgenesis, with Alzheimer's, with the progression of SARS, and with diabetes mellitus. The particular interest, my particular interest in it, is it was also associated with the first disease that we studied, which was a disease called sarcoidosis, a rare disease, but which turned out to be a very typical inflammatory disease. So if you want to study these diseases, you're going to be drawn inexorably towards a study of the gene for ACE, for the angiotensin converting enzyme. And it just so happens that a number of bacteria act on that gene. But in particular, lactobacillus and bifidobacteria, which are what we regard as being friendly bacteria, gut bacteria, that are in the yogurts and other supplements that some of us eat. These bacteria produce a number of peptides that downregulate the expression of ACE in the human body. So that is known, that very common bacteria, which nobody would argue are present in the human body, produce peptides that downregulate a gene that is a key gene in understanding the diseases for myocardial infarction, Alzheimer's, renal disease, progression of SARS, and diabetes. Therefore, if you're going to study those diseases, you have to study these organisms as well as the host organism, Homo sapiens. You're looking at a metagenome. You're looking at more than one genome that has to be examined. Similarly, we have another one down here, PTPN22, which is related to diabetes mellitus, rheumatoid arthritis, and lupus, SLE. That one is known to be upregulated as part of the innate immune responses, the innate immune system's response to mycobacteria, with the surge in latent tuberculosis, with our increasing knowledge of Mycobacterium avium, prevalence in the population. It's very important, if you're going to study these diseases, you need to study how that particular gene behaves in the environment that we live in If it's stimulated by mycobacteria, probably other bacterial species as well, that will be upregulated, and that's known. Once again, we're expanding the metagenome that we need to study when we think about disease. Back a number of years ago, I reported that sarcoidosis had succumbed to an antibacterial therapy based on a VDR agonist, that we're actually able to reverse that disease process. And in the subsequent 5 years, we put together an observational cohort of over 500 human subjects with a whole variety of different diagnoses. And we went on to demonstrate the reversibility of many autoimmune diagnoses. This is from a paper that was presented at the 6th International Congress on Autoimmunity just 2 months ago in Porto, in Portugal. And we have listed here a number of autoimmune diagnoses which responded to an antibacterial therapy which involved a VDR agonist, a small molecule called a VDR agonist. And we start at rheumatoid arthritis, Hashimoto's thyroiditis, uveitis, psoriasis, type 2 diabetes, Sjogren's, celiac, SLE, and go right down to the fewest number of cases which are in diabetes insipidus. In general, between 18 and 53 months of treatment, 81% of the cohort experienced reduced disease and symptoms with an antibacterial therapy involving a small molecule VDR agonist. But this is a surprising thing. Chronic fatigue syndrome, myalgic encephalomyelitis, osteoporosis, periodontal disease, cardiovascular disease, cognitive deficiencies. At Karolinska earlier this year, there was a conference on cognitive deficiencies in disease, and we gave papers showing both CFS and cognitive deficiencies were directly related to inflammatory diseases, and chemically some of the links. Obsessive compulsive disorder, bipolar and memory loss, things that we don't think to be associated with inflammation, these also disappeared as a chronic inflammatory condition, the primary inflammatory condition. Rheumatoid arthritis, SLE, whatever, disappeared. Now what we've done is we identified back around the turn of the century we identified that an intraphagocytic metagenomic microbiota, and we describe the method by which it evades the immune system, and that's where this small molecule comes in. So firstly, a number of groups in the past had identified that there were microbiota or communities of microbes which lived inside the cells. There are many studies, I'll show you one in a while. But what we did was we said, right, well, how do you deal with those? What do they do? How do they evade the immune system to persist inside phagocytes? I mean, the phagocytes are supposed to be the body's defense against a persistent infection. What we found was that the genomes in this metagenome, in the microbiota, the biofilm if you like, the genomes accumulate gradually during life, incrementally shutting down the innate immune system. And depending on the environment to which you are subjected, which pathogens you become subjected to during life, the metagenome will gradually build up during life, and eventually genes from the accumulated metagenome will determine a clinical disease symptomatology or just a disease of the aging. The microbiota is located in the cytoplasm of nucleated cells. Therefore, it has access to both the DNA transcription and to the protein translation machinery of Homo sapiens. In addition, and this is very important for those of you who are studying mutations, in addition, the host DNA repair mechanisms are susceptible to modification by junk from the metagenome. Well, what does a microbiota look like? This is light microscopy. This is a monocyte with a very highly infected cytoplasm. You can see the nucleus, there's the cytoplasm. And the cytoplasm has effectively exploded from expansion of the pathogens in it and thrown out dozens of these tiny, tiny transparent tubules which stain for bacterial DNA. And you see them along here. There's probably 20 submicroscopic bacterial tubules through there. There's infected cytoplasm and the nucleus. Look at the length of these tubules. This is about 20 cells long, this one. It's absolutely huge. This is optical microscopy of live blood, aged for between 6 and 36 hours on a microscope slide, untreated other than that. Just live blood aged on a microscope slide. It was taken on a chronic fatigue syndrome patient using a Bradford microscope by Dr. Andy Wright in Manchester. Now, there was a TEM study done, transmission electron microscopy, in 1989 at Columbia University by Emil Roscoe and his colleagues. And he looked at the phagocytes of all of the phagocytes actually from a number of diseases. He looked at Crohn's, he looked at juvenile rheumatoid arthritis, and he looked at sarcoidosis. And he took the phagocytes from the eyes of these patients and was therefore able to image them very well with TEM microscopy. And what he found was a number of artifacts inside the cytoplasm which stained for bacterial DNA. This is a stain. Unfortunately, this is 1989, and the TEM was all monochrome and non-enhanced at that point. And then there are also very small tubular structures, transparent tubular structures throughout the cytoplasm, not unlike what we were seeing on the other slide, but not nearly so heavily developed, not nearly so heavily parasitized. And here, of course, you've got the nucleus, and the nucleus itself is starting to break apart and become weak as well. This was from a juvenile rheumatoid arthritis patient. The important thing is to understand how the microbiota persists, because it's one thing to understand that a microbiota must be present, and that it is present, but you can't do anything about it until you understand how it evades the immune system. And that was the breakthrough we made back around 2003. And we looked at the nuclear receptors. The nuclear receptors are a family of very complex proteins that transcription factors that transcribe DNA for many of the key functions of the human body. For example, the progesterone receptor, the estrogen receptor, the androgen receptor, the glucocorticoid receptor are all nuclear receptors. And there's one of them called the VDR. The VDR nuclear receptor is very important for the innate immune system of Homo sapiens because it transcribes the genes for the cathelicidin and beta-defensin antimicrobial peptides. The primary way that phagocytes themselves protect themselves against pathogens is with cathelicidin, secondarily with beta-defensin. And those two genes and the transcription of those genes is essential to intraphagocytic innate immune defenses. The microbiota evades the human immune system by blocking DNA transcription via the VDR, which consequently blocks expression of these endogenous antimicrobials. In addition, the microbiota changes the expression of the 913 or more other genes that are transcribed by the VDR, and particularly including MTSS1. MTSS1 is the metastasis suppressor number 1. It gives rise to the protein called missing in metastasis. And when the bacteria knock out, when the microbiota knocks out the VDR, that is one of the other 913 important genes in the body that get knocked out as well. The homeostasis of other type 1 nuclear receptors is also indirectly upset by the pathogens. VDR, PXR, the pregnane xenobiotic receptor, glucocorticoid receptor, thyroid alpha-1 and thyroid beta-1 are all type 1 nuclear receptors which are upset when the VDR is knocked out by the microbiota. And in particular, when the glucocorticoid and thyroid receptors are knocked out, then we end up typically with diagnoses of hypothyroidism and adrenal insufficiency. We've demonstrated that both are reversible once the inflammatory disease can be reversed. Well, why has the microbiota been ignored? There are a couple of reasons. The first one is that VDR homology, the shape, the actual proteins, the amino acids that fit together to make the human VDR. VDR homology has evolved between the species in such a way that the VDR of Homo sapiens describes different genes from the VDR of other mammals. In particular, the VDR from the murine and canine genomes, which is what we've most generally studied when we've been studying diseases, doesn't transcribe cathelicidin or the defensins. So if the microbiota knocks out the VDR in the murine or canine genomes, it cannot persist. The phagocytes will get it because the cathelicidin and defensins are transcribed by different receptors in other genomes. It's only Homo sapiens and one or two of the other major primates that where the VDR transcribes cathelicidin. So a human metagenomic microbiota will not survive when it's transfected into a mouse. Koch's postulate fails. Different species and different mutations would be necessary if the microbiota was to knock out the different gene pathways needed for survival in a mouse. Now, of course, you would know from evolutionary statistics that the likelihood of a pathway developing in other mammals and other animals is high. And indeed, there was a conference a month ago from the American Society of Microbiology in San Diego, where a presentation was given by the Max Planck Institute in Germany, who have just found the same microbiota, the same biofilm inclusions in the phagocytes from worms that were recovered from barrier reefs, from coral reefs. So it's pretty certain that other species have developed, that microbiota have developed so that they can overcome the immune system of other species, but there is a different mechanism to the one that they use to overcome man. Now, secondly, the microbiota is only stable in vivo. It defies extraction using standard techniques. You saw what happened with that blood that was just aged under a slide. If you try and extract the cells that are infected disintegrate. It's very, very difficult to try and culture or identify these pathogens in vitro. But you can do it using DNA shotgun techniques. You can take the DNA from Homo sapiens and you can actually use shotgun techniques the same way as we decode the human genome or the bacterial genomes to figure out what genomes are there. And because we now have a roadmap of the 1,000 plus bacterial genomes, when we find DNA from those genomes in our human sample, we can say, aha, we are looking not only at human DNA in our shotgun sequencing, we are looking at bacterial DNA as well, and probably from X species, one of that 1,078 that we have defined. This is a study that was done by Dempsey et al. Actually used 16S RNA rather than shotgun, but it doesn't matter. They took infected prosthetic hip joints which were removed during revision arthroplasty, and they used ultrasound to get the biofilm off of the removed hip joints. And these are the species that they found in that biofilm, removed from people. Lysobacter, a typical bacterium, incidentally, you'd expect to find in a biofilm. At about 44% of the clones sequenced and about 60% of the clones analysed. Methylobacterium. Well, but Staphylococcus, well, you'd expect Staph aureus actually in biofilms, and it's there. Proteus, look at this, hydrothermal vent eubacterium. Hydrothermal vent eubacterium was present at about the same number of clones, a little bit higher number of clones of Staphylococcus. The iron oxidizing lithotroph, there are a number of species there that nobody dreamed existed in man until we were able to use molecular gene technologies to figure out, hey, what we're looking at is not Homo sapiens. It's not anything we've looked at before. The computer matches it up with the DNA from hydrothermal vent eubacterium. Until the genome was cracked, we only had the postulates of Koch as a guide. Koch, 1897, produced a set of postulates which defined when you could claim that an infection was a disease, that an infectious disease was present. They caused us to search for a singular pathogenic species because the postulates of Koch says one bug, one disease, and sidetrack science from understanding the horizontal transfer of DNA within the microbiota. Science became fixated on co-infections, things that the immune system couldn't knock out but which we could culture, which we could observe, and we missed the primary disease mechanism, the metagenome. The other reason that this has escaped study by science is that the VDR stands for vitamin D receptor. It is activated by a substance which we know as vitamin D, which is a transcriptional activator. It's a corticosteroid. Vitamin D is not a nutrient. There is a very complex control system by which 7-dehydrocholesterol is synthesized into 1,2,5-dihydroxyvitamin D, which then activates transcription by the VDR nuclear receptor. There are multiple feedback pathways. There is transcription of degrading enzymes. There is transrepression of an enhancing enzyme. And then via the PXR, the pregnane X receptor, other enzymes are affected. And then finally, this primary CYP27B1 second hydroxylation enzyme is activated by PKA, P300 CBP pathway. Very complex mechanism, which you would expect. It's at the heart of innate immunity. And a paper we published this year goes into the control system in some detail. And this is the interesting thing. Here is in silico technology. What we've done is we've taken the X-ray structure of the VDR, a number of VDRs actually, from different groups. And what we've done is we've docked the various types of vitamin D, various hydroxylations, into that receptor. Now, only one of them has the 1-alpha hydroxylation here, which is capable of activating transcription. But all of the others overlay the same region in the receptor. In other words, they are antagonists for that transcription process. Now, if we take the VDR agonist that we have found, which is a drug called olmesartan, we have olmesartan here in the human VDR, and this is a molecular dynamics emulation using a large computer array, computing real-time atomic force interactions. And of course, like all proteins, everything is in motion. There's total motion all the time. And here we have a rat VDR with the same drug in it. Now, what's particularly interesting is the orientation of the tetrazoles and the functioning of the drug is different in the two VDRs. If you look at that tetrazole orientation and that one, they're different. And in fact, there are 7 hydrogen bonds there, and there are only 5 hydrogen bonds in the VDR of a rat. So the drug itself doesn't behave the same way in the VDR of the rat versus the VDR of the man. Here is a basic problem that we have missed in our biological sciences. We have missed trying to go to the level of the gene and understand the difference between species. And we have assumed that any mammalian species is a reasonable emulation of the Homo sapiens genome. The other thing that you can do to go after the microbiota is to use low-dose antibiotics which block protein synthesis. The proteins that create the biofilms that protect the community can be attacked by a number of very simple antibiotics. This one's azithromycin. This is the 50S side of the ribosome with the protein translation area of the ribosome. And you can block the ribosome. The rate of bacterial death is controlled by inhibiting protein synthesis in the 70S bacterial ribosome using sub-inhibitory low doses of bacteriostatic antibiotics. And we're talking about very, very low doses here. With azithromycin, typically patients with inflammatory disease, once you activate the VDR, those patients can only withstand 1/10 the normal dose of something like azithromycin. One bacterium is weakened if just one antibiotic molecule is bound into one ribosome. So intermittent, low doses proportionately control the rate of bacterial death. We can actually proportionately control the rate of bacterial death by using the protein synthesis inhibitors. And that is just as well, because there is a phenomenon called immunopathology. Immunopathology is the damage done to the rest of the body when the immune system is doing its job. When the immune system is phagocytosing dead cells, phagocytosing pathogens, then it releases a cytokine storm, it releases chemokines, it activates the nuclear factor kappa B pathway, all of the normal ways that the body signals amongst itself that something is desperately wrong. When you start allowing the immune system to find the huge bacterial load we're carrying, 10 times the number of bacterial cells to the human cells is the NIH number, then the immunopathology can become life-threatening. The loss of cells, the cytokine storm, has to be controlled so it doesn't become life-threatening. So what we do is we use very low-dose antibiotics, sometimes just the immune system itself, to spread the therapy over many years. So there's one other way that we can visualize the metagenome, the way that the human experience is a metagenome, that the product of many, many genomes working together in tandem. Now, I pointed out earlier this E. coli glucose metabolism, and what I was pointing out to you there was that a lot of the products that are produced, fructose 6-phosphate for example, are very common in both E. coli and the human genome. Both produce those substrates, but there are some of the substrates, some of the proteins which are produced by E. coli, which I called junk earlier on in one of my other slides, but they are exudates, so they are byproducts of the bacterium itself working. And there are some of those that are unique, and we can use mass spectroscopy to image them. And at the Imperial College in London, that's what they've been doing. And they've come up with a concept called the metabolome, the human metabolome, or the spectrum of metabolites, which are associated with Homo sapiens. And for my final slide, I want to look at this particular image, which is from Dumas et al. In Analytical Chemistry, 2006. And what Jeremy and his colleagues at the Imperial College did here was they took those non-human metabolites that were present in urine, and they produced a method of displaying on a 2D surface the specific measures of those non-human metabolites in such a way that you could see that different populations were producing different metabolites. So the American population has different non-human metabolites in its urine to the Japanese, to the Chinese. Very large number, about 1,000 patients. And you can see that due to their environment, due to their food, due to everything that is associated with their metagenome, these three populations are different, and we can measure the way that their bodies are operating differently. One particular artifact which you can just see here, you can just see a violet number stuck here in the American group. There are actually 5 Japanese that moved to America. When they did that, their metabolome suddenly took on the American characteristics rather than their native Japanese characteristics. In other words, it's an environmental factors that drive our metabolome. So that's my final thought. Always remember the metagenome. The human experience is based on a metagenome. 10 times as many bacterial cells in the human body as human cells is the current estimate. Over the next 10 years, we'll be able to firm up exactly what that number is and exactly what the function of those, I hesitate to call them let me call them symbiotic cells rather than pathogenic cells. Thank you very much.
Thank you very much. I'm from India, IIT Kharagpur, and I'm working for a long time with the bacterial pathogens as well as the human genome relation. So my question is very relevant to MHC gene, major histocompatibility gene related to the bacterial pathogens. How they actually react, I don't understand actually even today that major histocompatibility gene with HLA function with pathogenic and non-pathogenic selectivity of the macros which are living in our system. And that is the queries that I wanted to ask you, whether you have something with relation to MHC gene and human pathogenicity as well as the microbiome infection, how to actually...
I don't have a micro answer, I have a macro answer. The answer is we're dealing with approximately 1,000 genes which are estimated to be in the human body compared to the 25,000 genes of Homo sapiens. When there is interaction, such as you would definitely have in the cytoplasm of nucleated cells, where there is interaction, the interaction is not on the scale of one protein affecting MHC. It's on a number of proteins and enzymes and, in fact, also small molecules from multiple species affecting MHC. Because MHC is so important to the adaptive immune response, you can expect that the pathogens are going to have developed some way of overcoming the adaptive immune response, and therefore some way of mutating, underexpressing, or whatever the MHC. So the macro answer is we don't know, but it makes sense. The micro answer is we don't know.
Actually, this is a very interesting and new phenomenon discovered that, like a computer, the IBM system is not compatible very with the Apple system. But now gradually, the two systems, they have some microprogram that can be integrated with one another. Would that be possible that the microorganism's genome can be sometimes in some mechanism that be dynamically integrate directly with the human genome?
Well, we have done that randomly. In the past there have been, for example, smallpox vaccination was very strong at one point in history. Polio vaccination with the different vaccines, for example, has been different for different generations of people living today. And so we have actually done that. We've also used BCG, a live vaccine, for example, in trying to combat tuberculosis. And we know BCG is related to causation of other diseases, sarcoidosis being one of them that's upregulated by BCG. So randomly we've sort of done that. But at this point we have no idea how to introduce another genome in order to try and make the metagenome on the whole positive rather than being negative. I mean, the phage therapy that was very popular in Russia and Eastern Europe, for example, is in many ways a way of trying to change the metagenome in such a way that it wasn't harmful. It wasn't making those harmful substances anymore. And in the short term, it worked reasonably well. But in the long term, it didn't produce very much extended longevity or any of the things that you would expect from it. Does that answer your question?
Yeah, sort of. Thank you. But you see, from the broad spectrum for retrovirus, the retrovirus genome integrated with human genome.
Yeah.
From time to time, like HIV.
Very common, yeah. In the telomeres, primarily starting into the telomeres and then working upwards. And if you look at these sicknesses, the inflammatory sicknesses, rheumatic diseases, the telomeres are different, they're ragged. There's something going on there that we don't understand yet. And that's where we've got to start looking.
Thank you. This the last question.
Very interesting for us who we do immunology. This is, for me, a completely novel concept. I mean, we're trying to use a lot of the TLR ligands as adjuvants. But on the other hand, you're telling us that we're full of bacteria all over, so we should be hypersensitive or sort of tolerant. But we still use the TLR ligands and induce very strong responses. So how you put this together? Because you show, I don't remember the gene that inside of our cells we have a protective gene not to be hypersensitive. So how you put this together with still responds very well like CpG, flagellin, whatever other TLR ligand?
Well, if you look at autoimmune disease in general, the focus in autoimmune disease research for the last 5 decades, last half century really, has been on the adaptive immune response, on the things we observe, the antibodies. And one of the diseases that I mentioned there, chronic fatigue syndrome, which is thought to be a neurological disease or just a psychosomatic disease by some physicians, but there are actually distinct antibodies which were identified in CFS patients. This was reported at the Portugal 6th International Congress in September. Because what happens is when the innate immune system fails, you get a cytokine cascade and a chemokine cascade which signals to the adaptive immune system that something is wrong. And it tries to figure out what's wrong, it tries to take the fragments of whatever's available, whatever DNA has been left after phagocytosis, and figure out what to do with it. And when we think that we're producing antibodies to the human body itself, we're actually producing antibodies that is the adaptive immune system's best guess at what the pathogen looked like. But because we haven't identified the pathogens enough yet at the level of the gene, at the level of the DNA, we don't realize that we're looking at a pathogen. You would know that antibodies, for example, are very non-specific. A typical antibody will react to anything from a range of small molecules right up to a number of larger proteins. Some are very specific, but very few are very specific. So if you look at the adaptive immune system, it is downstream from the innate immune system failure. And we've just always assumed that the innate immune system just worked. And it hasn't been just working. It's been gradually getting worse and worse in its operation, particularly during the last century. Although these diseases have been around, the Neolithic Iceman that was dug up in Austria, Italy, a decade ago, for example, shows sign of stroke and also arthritis, which are the same diseases. So it looks as though a metagenome has been around for quite a long time, just changing as each successive generation comes along.
Thank you, Dr. Mashu.